Hi everyone, my name is Lina Moammar. I am situated in Orange County, California, and today I'll be presenting to you my slides that I have titled New Strategies for Early Detection of Alzheimer's Disease. A little background about myself is that I started this research when I was in my junior year of high school, and I was able to publish it by my senior year of high school to the Curio Academic Journal. I am now a rising neuroscience major this school year, and I am very excited to be sharing this research with you all today. So, of course, every scientist and every individual has a backstory to the things that they're passionate about. So, for me, a few years back, my grandmother was actually taken away from us by Alzheimer's disease. And a few years later, we found out that my grandfather was also diagnosed with Alzheimer's disease. So the overlap of these two events made it a natural fit for me to explore the question of whether we could identify Alzheimer's disease before its debilitating symptoms manifest. Can you imagine a promising answer to that question? What would that look like exactly? So, early detection could offer families more time to create cherished memories with their loved ones before the disease takes its toll, as well as opening doors to interventions that slow down or even halt the progression of the disease altogether, potentially offering families what mine could not have. But to do this, we need to understand more about the Alzheimer's brain. So there's one noticeable difference between these images. One distinct difference between the healthy brain and the brain with Alzheimer's disease is actually the size of the brain. But we can't measure that very easily. I mean, we can measure it, but it's not cheap, it's not easy to do, and it's also not accessible to everybody. What's available right now to diagnose Alzheimer's disease relies on conventional imaging such as a PET scan. This would be able to give an image of the brain to determine the size and then diagnose Alzheimer's disease. However, it's expensive, it's not practical, and it's almost kind of claustrophobic. So there has to be a better way. What is different about Alzheimer's patients? Perhaps something we could exploit that is meaningful and detectable, and that's not expensive or claustrophobic or scarcely available to all grandmothers. Now, if we return back to our two patients, there actually is another difference. It's the amount of a neurotransmitter called acetylcholine. A neurotransmitter is a chemical messenger that allows the brain to function. Acetylcholine plays a pivotal role in memory and cognitive function. When there's a normal amount, the brain functions properly. When there's less, memory and cognitive function are both impaired. In fact, it's the absence of this neurotransmitter that is closely linked to Alzheimer's disease. If we could measure acetylcholine concentrations, we could monitor for early warning signals of Alzheimer's disease. There's actually research that is exploring this very approach, but what is missing is a sensitive method for detecting acetylcholine levels. Instead of the imaging, this new diagnostic would screen patients using a blood test, which is familiar to everybody. It's cheap, accessible, and minimally invasive. The blood samples would be analyzed for acetylcholine levels as denoted by the black dots. The next question is how to detect acetylcholine within the blood sample without leading to false positives or misleading concentrations. We need something that will bind to the acetylcholine in the blood sample and nothing else. We need a way to accurately report on that binding. Through extensive literature review that was guided by my mentor, I explored the potential of polymers and their capacity to bind with acetylcholine. This is where my AP Chem knowledge really helped. A polymer is a chemical that has repeating units, much like a string of pearls, where each individual pearl is a unit and the string itself is actually the polymer. Why polymers? These materials are versatile and can be engineered to have specific properties. The challenge was to find polymers that could effectively bind to the acetylcholine and be used to determine acetylcholine concentrations. I identified three promising candidates: itaconic acid, acrylamide, and methacrylamide. Now, there's a lot of details that go with identifying compatibility between these molecules. But for reference, this is the structure of itaconic acid, and its carboxylic groups are pictured in red. These polymers have their amide groups pictured in red as well. Acrylamide also has a vinyl group, and methacrylamide has a methyl group that has binding properties to the acetylcholine. In summary, these properties have a high binding affinity with the acetylcholine, which allows for the formation of what's called hydrogen bonds between these polymers and the acetylcholine. But how do we measure these binding interactions accurately? We need to find a way to report on the binding event itself. So here's how we can do that. When ACH and the polymer come together, they could, I guess you could say, click in place, as this picture shows. But we can't actually use a clicking sound to report on a binding event because chemicals don't make sounds when they bind together. Instead, we could use what's called a biosensor that lights up when binding occurs. Here's how the idea would look in the lab. A fluorescent tag would be attached to the polymer. The polymer tag combination is then immobilized or placed on a surface. Then the blood sample would be passed over the surface, and any acetylcholine present would bind to the blood sample as shown to the polymer. The fluorescence is measured, and then the amount of acetylcholine would be detected. By now, you might be wondering how this could actually translate into real-world impact. Imagine a medical practice that is equipped with a tool capable of tracking acetylcholine levels, potentially identifying Alzheimer's-related deficiencies before symptoms occur. Currently, all methods of diagnosis happens when the disease has already advanced and taken its toll, as the yellow arrow depicts. Imagine being able to diagnose Alzheimer's-related deficiencies before the symptoms take place. Could help us buy time. How many people are we really talking about, though? Today, there are 6.5 million Americans older than 65 years old who have Alzheimer's disease. Researchers predict that this number will grow to 14 million Americans by the year 2060. We simply do not have the tools to detect or slow or treat this disease. What is the burden on the US healthcare system? The total payments for healthcare, long-term care, and hospice in the United States for people with Alzheimer's and other dementias are projected to increase from $290 billion to $1.1 trillion in 2050. So it's not getting any better. What are the treatments currently available for Alzheimer's patients? Well, nothing. There's only one, and this treats the behavioral side effects of Alzheimer's by stabilizing the existing acetylcholine that is circulating in the body. It does not slow down the progression of this disease. We really do not know what to do. It's all the more important to monitor the progression of Alzheimer's disease so that we can identify different milestones that could be targeted to slow down the progression. If we don't know when it starts to show up, then how will we develop effective treatment plans? Then healthcare could be offering individuals and their families precious time to take proactive steps to slow down the disease or even avoid the progression altogether, if we had the right tools. So what does this ultimately mean in the larger conversation around Alzheimer's disease? It signifies the possibility of shifting from reactive management to proactive prevention. So now, where do we go from here? These ideas must be tested. It's important that we validate and solidify the potential use of these polymers and detection methods in order to move forward. By replicating and expanding on the potential for these polymers and their detection methods, we can cement the reliability and accuracy of this proposed methodology. Finally, if we engage individuals at risk of Alzheimer's through in-lab studies, these real-world insights could actually offer compelling data that would strengthen the link between acetylcholine deficiencies and Alzheimer's progression, and of course, giving families the extra time that they need with their loved ones. So I have contributed something to solving this problem in the matter of 9 months. I've watched myself evolve and understand how to conduct research and connect with something so special to me. And I hope this I can take this with me as I enter university. Of course, you can also do the same thing and participate in using your interests to solve a real-world problem. As Gandhi said, "Be the change you wish to see in the world." What can you do to help? Whether it be contributing to Alzheimer's research or finding something you're passionate about and going further with it, there's always something you can get involved in with the things that you care about. You can also scan this QR code to the Alzheimer's Association, and they have resources on recent Alzheimer's uh, research progression, as well as ways to advocate for legislation to fund this research. As always, thank you for listening.